Extreme and unusual habitats. These two examples here, these are lakes in the dry valleys of Antarctica that are permanently ice covered. This is Mona Lake. It's a very saline lake in California, and we'll talk about a whole series of extreme and unusual environments. Not commonly covered in a lot of aquatic ecology classes, but interesting to me, so I'm going to make you learn about them. The habitats we'll talk about: saline lakes, hot springs, cold habitats, temporary waters, ultra oligotrophic, deep subsurface and groundwater, and then at the surface layer of water. So I'll talk first about adaptations to thermal extremes. And this has importance both in extreme environments and also just how organisms adapt to different temperatures, or why some species do better at one temperature and others do better at different temperatures. Why does a trout do better at cold water and bass do better in hot water, warm water, for example? So there's a number of things that you can do to change your ability to deal with the environment. And one of the things that we need to think about is how proteins work, how DNA and RNA work, and how lipids work to make cells be functional. Life is a very strange thing because it is perched between order and disorder. We can take an example of lipid bilayer to be functional. So the lipid needs to be dissolved, the lipid tails need to be dissolved in each other, and the charged phospho part on the outside. And then there's proteins in that, and those proteins need to be able to swim around in it. But the temperature can't be so high that that membrane breaks apart. And it can't be so low that the membrane's essentially solid. So it's semi-fluid, right? It's organized, but it's fluid. Same thing with proteins. Proteins essentially dissolve their hydrophobic parts on the inside. They have their charged parts on the outside. They have ways to cross-link with sulfur bonds. But what they're doing as enzymes, when they're acting as enzymes, they have an active site. And it has to be a very specific configuration to exactly match the molecule that it's supposed to be reacting with. Either two molecules that are squooshing together, right, to create a reaction, or ripping apart. But when it's doing that, it's translating thermal energy, right, into chemical energy. So we could think of, say, something that is nitrogen-fixing bacterium, nitrogenase. That enzyme has to match that nitrogen, right? And then it needs to pull those triple bonds apart. So it needs to have enough thermal energy to basically do the work of pulling those bonds apart, but not so much thermal energy that the structure is out of whack and that it won't recognize that nitrogen molecule. So again, it's on this sort of fine line between solid and liquid, between order and disorder. And DNA and RNA have the same thing. If it's too cold, you can't basically replicate DNA and RNA. If it's too hot, they just fall apart and they lose their structure. We already talked about that with respect to the ribosomal RNA, in that the ribosomal RNA has parts of the molecule that are very essential to the structure and the activity of that molecule. Other parts are just essential to structure in that they base pair with each other to hold structure, but they don't actually have enzymatic activity. Yet other parts are just loops to get the molecule around, and they have really no importance whatsoever other than they're just kind of a spacer. Right? So we talked about how structure can matter there. So we can think about an example of why, if we had high temperatures, what you'd have to do to make yourself do okay at high temperature. Your DNA can't be denatured. It can't fall apart, right? So how do you stabilize it? You can increase the number of CG pairs. Why does that increase stability?
Because DNA pairs denature faster.
Because What's the difference?
They only have like 1 or 2 bonds, right?
2 versus 3 hydrogen bonds, right? So they tend to, so within a group, if you can get more CG pairs, you can stabilize your DNA. You can also put proteins around them that stabilize them, that that hold them in place. RNA has similar things. The different types of RNA have to be able to bind to themselves to create tertiary structure. And to do that, you can have more extensive regions of base pairing in areas that need to hold the conformation. So what happens is these thermophilic organisms have constraints that are built into their DNA and their RNA. And if you put them in a cold environment, all of a sudden those molecules freeze up, right? So they don't work. Likewise, if you take a cold adapted organism and warm it up, it's not going to do so well. Membranes. You can think of some examples of membranes and lipids that are adapted to different temperatures. So why is it that when you go into your kitchen and you have butter, it's solid, yet canola oil is liquid? In a general sense of how the organisms that created it, that make those compounds, are adapted to their environment. And ultimately the answer is evolution, of course, right? But where does butter come from? Nobody knows where butter comes from? Cows, good. And where does canola oil come from?
Canola.
Canolas, which are plants, right? So what is it different between a cow and a plant as far as thermal regime?
Cows are warm, plants aren't.
Cows are warm-blooded, right? They're always running at about our temperature, right? Plants, if they're going to be going now, they're going to have to be running at, close to 0 degrees C if they weren't going to still be active. Generally, they're colder than animals are, so they need to keep their lipids fluid. If you cool us down, right, to 5 degrees C, we're dead because our lipids solidify. They're set to be fluid at the temperature they're set. And we do this mainly by increasing saturation, branching and laying. So that's also why eating a lot of butter and animal fat isn't great for you, because it tends to solidify at higher temperatures and coat your arteries and such, right? Whereas plant oils tend not to do that so much. They tend to be more healthy for your body as well. There's practical application of this. Also, we talked a little bit about how proteins need to maintain their active sites, and they are using that thermal energy to yank things apart or stick them back together. So they need to have the right shape, but they need to be able to wiggle around a little bit too. How do they make themselves more stable? There's beta sheets and alpha helix. They have a more extensive hydrophobic core and higher external charge. Right? So if you remember, proteins can bond to themselves in sheets, essentially hydrogen bond to themselves in sheets. They can also form helices in a similar fashion to DNA or an RNA. And because the amino acids have different polarities, some of them are very hydrophobic. If you're dissolved in water, they'll dissolve in each other and really resist changing, coming in contact with that water outside, and all the charged amino acids will be on the outside. So the more you shield the inside and the outside from hydrophilic and hydrophobic there, that solidifies things. So this is thermal stability. Salt is an interesting thing that needs to be dealt with metabolically. If you have very high salt concentrations, water is actually a limiting factor. And that's because if you remember from your chemistry, the chemicals have these ions, chemical molecular ions have waters of hydration. So, you know, you might have like 5 waters in the formula of a chemical compound. Those means that they're complexing pretty closely with water. So if you have a whole high concentration of that salt, it's all complexed up with the salt, and biologically it's difficult to get to it. In addition, if there's really high concentrations, we talk about how this high external charge here is holding the conformation, but if you have a lot of other ions out there, it tends to interfere with their ability to use that charging and hydrogen bonding to bond with themselves. So you have this ionic shielding. Complex animals pump out salt, right? So us, fish, we just regulate our salt at some concentration. The extreme halophilic and archaea bacteria and archaea are isotonic. That is, their internal and external salt concentrations are the same, and so they've adapted to dealing with those super high concentrations. Some organisms will use glycerol to remain isotonic, so they don't have to deal with the high salt concentrations. If you remember back in Principles of Biology, you did that thing where you took blood cells and you put them in a salt solution and you look under the microscope and they just explode, right? Because they're trying to get to the same concentration of, no, I'm sorry, you put them in deionized water and they explode. You put them in salt and they crinkle up, right, as they're trying to get as concentrated inside as outside. And so if you have a high concentration of glycerol, you'll be able to maintain your cell shape. And in dry environments, you have pretty similar adaptations to organisms as you do to salt. They must be able to withstand desiccation, and then oftentimes as water dries down, it becomes more and more salty. So you run across the same problem. So now that's sort of setting up the molecular basis of it all, biochemical basis, and then we'll move into the individual habitats themselves. So the first one is saline lakes. Saline lakes occur in closed basins. And if there's going to be water there in a closed basin, it doesn't have anywhere to flow out, so it's got to leave by evaporating. And if there's going to be water there and it's going to be salty, the long-term precipitation and evaporation have to balance each other. So the water will come in out of the watershed, having weathered the salts away from the terrestrial surface, and bring those salts into the lake, and then as it evaporates off, the salts remain behind, and so it gets saltier and saltier over time. So many desert areas or dry areas on Earth are saline, and some of the largest lakes on Earth are saline lakes. Because there's this continuous evaporation, the chemistry is really dependent upon what the chemistry is of the soils and rocks surrounding those lakes. Right? So you can imagine if you have sodium chloride as the predominant salt, as opposed to sodium bicarbonate, you'll move towards a system that's dominated by sodium chloride as opposed to bicarbonate. And then your pH and your bicarbonate will be high, your pH and your sodium chloride will be more variable depending upon what's happening there. As you increase salinity, actually oxygen concentration gets lower. And so you might actually, in very hypersaline situations, only have organisms that can deal with low oxygen concentrations. Then there's groups of organisms in these extreme environments that are more tolerant than fish are and able to survive. So brine shrimp is an example of one that's commonly seen in saline waters, that if the fish get in, they're out. Any of you ever have sea monkeys when you were kids? Yeah. So that's just brine shrimp. They're just a nice desiccation-resistant eggs. You throw them in salty water and they pop out and go around. If you got algae in there, they'll live for a while, maybe even have a couple of generations if you're good at it. Yeah, so you can get these sort of interesting dynamics. One of the saline lakes in Oregon, Abert, there's a spring that came out in the middle of the lake, and this guy made his whole living going out there with these fine nets and setting them up in front of the spring, and it pushed the brine shrimp onto that net, and he'd pull the nets out and scrape them, and he had a truck that had a freezer truck, and he'd throw them in there, take them down to California to sell them to the pet food stores for fish food. But they came into a wet period of time, and more water came into that lake. And what do you think happened to the salinity as the lake level went up? It decreased, right? And it decreased to the point where the fish could actually survive in there. So the fish are all in the river, freshwater fish are all in the rivers. They came in and had an absolute feast and just knocked the brine shrimp concentrations down to almost nothing. This guy couldn't make a living doing it anymore, so he left. And then when the lake got saline again, he never came back because he was sick of being out in the middle of the desert, scooping up brine shrimp all the time. So one of the big conservation issues is saline lakes. The Aral Sea was the 4th largest lake in the world, but irrigation from the watershed above has decreased the area of the lake by half. And these dust storms are because of this high concentration of salt in the dust, it's really actually not good for you. So it's caused some serious health problems in the region, as well as there being particular species that are adapted to these saline systems and have become endangered or threatened because of the loss of these habitats. So this is another area where the conservation has really kind of come down. Another area in the United States that has a very large saline lake is the Great Salt Lake in Utah, which at one point covered perhaps over a third of the state. And that lake was then called Lake Bonneville, and it actually was a wetter period of time, and there was the water would come in there and it would spill out to the north and drain into the Columbia River basin. So it was a freshwater lake, and it laid down these sediments all over the place. And as the precipitation dropped, then it dropped below that outlet and became an evaporative system and got more and more concentrated. It left, in some areas, it just completely disappeared and left the salt. So that's the Bonneville Salt Flats where they test the high-speed cars and all that kind of stuff. It's that flat because it was the bottom of the lake, right? And then the Great Salt Lake that's left behind, has then protozoa, brine flies, but no fish. It's too saline. The other thing that's interesting with these saline lakes that people oftentimes do is they use them as a source of salt. And so they'll pump the water into a series of ponds and evaporate it even further. And as you evaporate it and make it more and more concentrated, different salts become supersaturated and fall out of solution. And so the salts themselves will give them these different colors, but also there are bacteria that can live. Some of the archaea and the bacteria can live in hypersaline conditions. And it turns out that the bacterial composition helps precipitate those salts because it forms a nucleus for the salt crystal to start forming. So, yeah.
So, could you stock ocean fish in those lakes?
Yeah, so it's happened. And probably the biggest example is the Salton Sea, which is actually an area that water sort of spilled into and then became saline in California, and then they actually put in marine fish in their stock of marine fish. The salinity started swinging enough that they weren't real successful at it, but it's a big problem as far as trying to maintain fish species. You have to be in kind of a narrow window. The marine fish have not evolved to have to tolerate very wide ranges of salinity, especially on the high side. The estuarine ones have evolved to get lower salinity down to fresh water, right, and back and forth. So they do okay if you're kind of have a moderate salinity, but yeah, you can do that. So they are used for fish culture, for marine fish culture in areas that have saline lakes. Good question. Bronson, you have a question?
The one in Utah, you said there's no fish.
The Great Salt Lake doesn't have any fish in it.
They haven't attempted to do ocean fish in there.
It's too salty for any fish. So this is just a picture of your sea monkey with the eggs on the back of it, and your brine shrimp. It's a crustacean, remember. And this is just an idea of sort of the upper levels of salinity tolerance in percentages of a variety of different organisms. So this gets back to Bronson's question. Fish sort of have the lowest of all these groups of organisms, that the most tolerant fish can deal with up to 11%. And the Great Salt Lake is running somewhere up, up in the 20s at times, depending upon what time of year it is, or even up in the 30s. So we can see, in general, there's a trend here with extreme habitats, is that as you get more and more extreme, first the multicellular organisms drop out, then you get to basically bacteria and archaea at the most extreme levels. And that's sort of the general trend, whether you're talking about salinity, high temperatures, pH, any of those potential extremes.